Cell division is important for growth and development of any organism. In fact, our life starts with a fertilization event where sperm is fertilizing the ovum, and successive round of cell division ensures that growth takes place and makes us what we are right now. But have you ever wondered that how these sperms and ovums, that means the gametes which leads to the fertilization, how they are produced? It turns out the sperm and the ovum are produced by a special type of cell division paradigm, which is known as meiosis. It takes place in the gonads. In case of the male, it takes place in the seminiferous tubule. Actually, the germ mother cell gives rise to the gametes in this process. Similarly, for females, it takes place in the ovary and which give rise to the oocytes. And this process is super important in terms of creating genetic diversity among the offsprings. That is why we really need to understand this process and the bits and pieces and the mechanisms underlying this process. In meiosis, DNA replication takes place once and it follows by two rounds of chromosome segregation, termed as meiosis 1 and meiosis 2, which ultimately leads to production of four haploid cells from a diploid mother cell. And these haploid cells are mostly the gametes, and they have some alteration in terms of their gene and the chromosome assortment, which we would be discussing in this whole lecture. So everything starts in the interphase. Just like a normal cell undergoing mitosis have to have an interphase, here the germ mother cells also undergoes interphase, where in the G1 phase cell organelles duplicate and start to increase in number, whereas centrosome duplication also takes place in G1s boundary. Like mitosis, in the first S phase of the meiotic division, DNA replication takes place, right? And then what happens in the prophase, the crossing over of chromosomes takes place. Crossing over takes place between non-sister chromatids of the homologous chromosome. This crossing over event is so important in terms of creating genetic diversity. After that, in the metaphase, these chromosomes, which are now termed as bivalents or tetrads because they have now four chromatids, they align across the metaphage plate. And microtubules, which are coming from the opposite sides, attach to their kinetochore, and they are ready to be pulled to the both sides. Now these bivalents actually separates and migrate towards the pole in the phase of anaphase 1 of meiosis 1, followed by a telophase event. This is pretty quick phase while the nuclear membrane reappears and the chromosomes again gets decondensed. So that is the end of meiosis 1, and after that meiosis phase 2 would take place. In the meiosis 2 or the prophase 2, similar event takes place, but in this phase no crossing over takes place. In beyond this phase, these chromosomes get aligned into the metaphase plate. Now the microtubules are capturing them from both the sides. Remember, in the metaphase 1 they were only captured from one side of one particular bivalent. Now here, due to equal tension, they are separated from each other and both migrates towards the opposite pole in the anaphase 2 of meiosis, which ultimately leads to segregation in telophase 2 and reappearance of the nucleus in telophase 2. That leads to production of four cells which are haploid in nature from a germ mother cell. Now let's look at the process in a bit more details. The first phase of the prophase 1 is basically leptotene. Leptotene stands for thin thread. At this stage, the chromosome is actually individualized, or it can be visualized under a microscope just like a thread. That is why this stage is named so. Leptotene is very short in terms of duration, and in this particular stage, progressive condensation of the chromosome and the coiling of the chromosomes takes place. Just after leptotene comes zygotene. In zygotene, the chromosomes approximately line up into homologous pairs. So we know for each chromosome, for example chromosome 2, there would be two homologous pairs, right? And these pairs are lining up each other in a line, and they are forming a presynaptosomal complex, a synaptonymal complex. And at the end of this phase comes Pacoitin, where the key event is the homologous recombination and the strand exchange between the two chromosomes. And the strand exchange takes place between the non-sister chromatids of the two homologous chromosomes. Before that, before the exchange takes place, there is a X-like structure formed which is known as the chiasmata or the junction. This junction is later on dissolved in diplotin stage, where the strand exchange is pretty much complete. Followed by diplotin, there is a very short phase which is almost overlapping with the pre-metaphase. This is known as diakinesis, which we are not discussing here. So just to summarize the events of the meiotic prophase is like in leptotene stage, the chromosomes start condensing and becomes individualized. In zygotene, chromosomes are aligning in homologous pairs, where in Pacoitin, the main event of crossing over takes place, followed by diplotin, where the chromosomes especially exchange their strands and the synaptosomal complex is actually degraded. Now, just like a mitotic prophase, in the meiotic prophase as well, cyclin B and CDK1 takes important role to phosphorylate nuclear pores and degrade that. Not only that, the nucleus is actually supported by the lamin, which are the intermediate filaments, and lamin has specific domain structure. What happens is this lamin is getting phosphorylated by cyclin B and CDK1, leads to their dissociation. When the lamins are dissociated, the structural framework is gone. It would ultimately lead to a collapse of the nuclear membrane. That is pretty much common in case of both meiotic and mitotic prophase. And ultimately, these spindle apparatus would move to the opposite poles because they had to pull these bivalents from both the sides. Now, it's important to note at meiosis 1, at the metaphase 1, the chromosomes are attached from the both sides of the bivalent. But for each particular chromosome, it is not attaching from both the sides. This whole organization is known as a tetrad, where there are 4 chromatids. Now in anaphase, these bivalents are actually separated. One is going from one side of the pole and another is going other side. However, in meiosis 2, in the metaphase, the microtubules are exactly attached to the opposite side, and it is ensured that equal tension is experienced in both the sides. That is why when in anaphase 2 these microtubules are shortening, then the chromatids are individualized and separated and pulled towards two opposite poles. Ultimately, it leads to 4 haploid cells with a slightly different genomic configuration. And that is the basis of all sort of diversity. But have you ever wondered why meiosis prophase is very different from mitosis? How it evolved during the evolutionary timeline, and why it is only and selective towards the gametes, not the somatic cells? these kind of discussions are beyond the scope of this particular lecture. We would be talking about this kind of discussion later in some other videos. But for now, if you like my video, give it a quick thumbs up. Don't forget to like, share, and subscribe. And I'm also available in Unacademy platform, which is India's largest online learning platform. Here I interact live with the people, so you can always join Unacademy and take a plus subscription, which is super cheap, and use my referral code AP10 to get a 10% discount. I hope to see you in any of these platforms. Thanks for listening.